Okay, so now we'll go to the good stuff. Prokaryotic, replication, transcription, and translation. Or 3 steps. So, overview of this, what is replication? Basically, replication is duplicating DNA. Usually, DNA is read from the 3 to 5 because it's synthesized from the 5 to 3. There's specific things that you would need for replication. And those are called, it's a complex called a replisome, which contains a DNA polymerase. It's the one that synthesizes the DNA, and which also always concurs from the 5 to 3 direction. And you also have the helicases that kind of untangles the DNA, like right ahead of the polymerase. And you also have the gyrase, which is it breaks down the strand of DNA and it gets like the 2 strands of the DNA untwisted. And it's a target site of antibiotic medicines, so only prokaryotes would have it. Of course, you have the DNA ligase, which basically joins the 2 DNA fragments together, forming a covalent bond, the sugar phosphate residues of adjacent nucleotides. And of course, you have a primer, which is ribonucleic acid. DNA polymerase can add nucleotides. And so actually, for bacterial DNA, it goes in a bidirectional. So there's the leading strand and there's a lagging strand. And the lagging strand is actually kind of goes backward because the directionality of it doesn't allow it to like continuously be synthesized. So I wish I could like insert pictures in here. That would be really cool. But you're just gonna imagine this. Basically, the leading strand goes from from like naturally from a 3 to 5, so you can just straight on go to 5 to 3. So this end will be like the 3, and I can do like 5 to 3. But unfortunately, the lagging strand, it basically goes from this is like the 5 to 3 end. So I had to go like one little segment here, I break off, one little segment here, break off, one little segment here, I break off. So all these little segments are called Okazaki fragments. And basically that's pretty much replication. If you want to know about transcription, here it is. Transcription is basically turning the DNA to RNA. So you know how you have adenine paired with thymine, and then cytosine paired with guanine. The thing here is in transcription, thymine is still paired with adenine, but adenine is now paired with uracil, which is a DNA. It's an RNA special. The DNA doesn't have a uracil. So therefore, you have a transcription. And what are the transcription things that you need? You need a sigma factor. You need an RNA polymerase again. You need a promoter, and you need a terminator. So sigma factor is basically it recognizes the promoter region. And okay, so it allows the cell to specialize, to kind of synthesize different strands at different times. And the promoter is the sequence that RNA polymerase binds. So there's a special sequence that you would have to do. I believe that's it. Oh, and translation, very important. Translation is a pretty complicated process. Basically, you transfer the RNA So like 3, there's like for example, can't think of an example right now, but you know, you would have like GZC. It stands for a particular type of protein. Oh no, not protein, amino acid. So you just continue on like that. Anyways, so that's the basic things of, you know, prokaryote, replication, transcription, translation. More details later.